So hello again. We're back with our penultimate piece of our module on nucleic acid structure, DNA replication, and chromosome structure. In this video, we are going to take a look at DNA replication. Now, this is complicated to describe in words and static images, so it is absolutely critical that you watch some of the videos that have been provided. Take a look at some videos, and that will help you understand what's going on here. Okay, so let's start off pretty fundamental. The point of DNA replication is to take one DNA molecule and make two exact duplicates of that. Now, yes, this is not an entirely accurate process, so there will be the introduction of some changes to the DNA sequence when this happens, and that is one of the ways in which mutations occur. And so this is an important way to introduce genetic variability into populations. We're not going to focus on that. We're going to talk about the mechanism as though it is completely accurate. It is highly accurate because the mutation rate needs to stay very low. In fact, it stays around 10 to the minus 6 mutations per cell division. So very, very accurate. So we've got a DNA molecule at the top here in red. And what we're going to do is we're going to use that to make two new molecules which are identical in sequence. So let's take a look at our sequences. C base paired with G, AT, CG, TA, GC. So what we're going to do is pull those two strands apart. We pull those two strands of DNA apart, and we use the information in one strand to make a new strand of DNA. And then we use the other strand to make a second new strand. And so the new strands here are shown in blue. Let me grab my pen real quick so I can mark things up. Okay, here we go. So here is the parental molecule. So here's the parental molecule. This is the molecule we're going to start with. We're going to pull the two strands apart. So here's the strand on the left, and then over here is the strand that we pull apart on the right. So note the polarities. So at the top here, we've got a 5' and down to a 3'. And remember, DNA is anti-parallel, so the strand, the other strand is going to run in the opposite direction. We'll come back to this important point in a moment. So here is our template parental strand. I say template because we're going to use the information in it to make a new strand of DNA. And here's the new strand that's been made in blue. And the resulting molecule, you'll see, has got exactly the same DNA sequence as what we started with in the parental molecule. And you'll see that the new strand that's made runs in the opposite direction to the parental strand. So it's already got pretty complicated, even though I said we're going to start off pretty simple. The reason this gets complicated and it gets frustrating and hard to follow is because of the anti-parallel nature of DNA. Remember, that's where one strand runs in one direction and the other strand runs chemically in the other direction when we consider one end is 5' and one end is 3'. Exactly the same thing happens over here on the right. The information in the red parental strand, excuse me, is used to make a second strand, the daughter strand. And we end up with two new molecules which in an ideal world are exactly the same as what we started with. Okay, let's make it a little bit more complicated. We've got to think about this happening when the two strands are wrapped around one another to form this structure, the double helix. So what we have to do is pull the two strands apart in the helix. And so what's happening here is that the two strands are being pulled apart. And the point where they're pulled apart and all the DNA synthesis happens is this structure here called the replication fork, because the DNA is basically forming, it runs like this, and then the two strands get pulled apart. So here is one of the parental strands in red, and then this new strand is being formed, the blue strand, that's the daughter strand, and as they form, they're obviously going to rewind to form the double helix. And so let's look at this strand over here on the right to start with. So here is the parental strand in red running around like this. There's the parental strand, and what we're going to do is we're going to make this new daughter strand. So here we've got to look at the polarities of the molecules. So the daughter strand, the parental strand on the right is 3' at this end and is 5' at this end. So that means that this end of the new daughter strand is the 3' end. And this is the 5' end. So let's just see if I can label that 5' so we keep it in our minds. Not very good 5, but you can make it out as 5. So here is the 5' end of the new strand. And it's being made towards its 3' end. So the nucleotides that are being added this G that's coming in here to match this C, and this C to match this G here, when they come in, they're added to this 3' end here. And that must be 3' because the other end, like we said, is the 5' end. And so this piece of DNA is being made so that it moves away from the replication fork. So chemically it's being made in this direction. Put an arrow on here, it's being made in this direction away from the replication fork. Let's see what's going on the other template strand. So the other template strand, here at this end it is 5', and down here it is 3'. So here is the first rule about DNA replication. All new DNA synthesis proceeds in a 5' to 3' directions. What that means is you must always add a new nucleotide onto the 3' end of a strand of DNA. You can't add them on the 5' end because of chemical and thermodynamic reasons. You must always add them on the 3' end. So here is the 3' end of the template strand. That means that this end of the daughter strand, the newly synthesized strand, this must be the 5' end. This is slightly better 5 because it's a little bit curvy. So this is the 5' end, which means that this end here is the 3' end, and that's where we've got to be adding the nucleotide. So in comes the nucleotide here. It's going to match this G, and the next C that comes in is matching the next G in the template. And so these are being added to the 3' end. We always go in the 5' to 3' direction. So the synthesis of this strand is in this direction. So what you now see is we've got one new strand being made away from the replication fork and the other new strand being made towards the replication fork. So you've got two things that they're going in opposite directions. This is going to cause some real problems when we look at the actual mechanics of the enzymes and the process of this happening. So I've set up the problem we need to solve. Let's back off a little bit and look at the mechanics a little bit more in a little bit more detail. So we've got a piece of DNA that we want to replicate. The first thing we've got to do is pull the two strands apart so we can get at the information in the templates. We need to be able to read the sequence of A's, T's, C's, and G's. So that means we've got to pull the two strands apart so we can then put the new nucleotides in. That happens at a point on the DNA called the origin of replication. Now, in bacteria and prokaryotes, their chromosomes are usually circular and they are much smaller than in eukaryotes, and so they often only have one origin of replication. Eukaryote genomes are huge though, much, much bigger, and so they must have multiple origins of replication. If they didn't, it would take them forever to replicate. So we have multiple origins of replication. We're just looking at one. And so at the origin of replication, a bunch of enzymes get together. They recognize that sequence of DNA at the origin of replication, grab the two DNA strands, and some energy is expended, and we pull the two strands apart. So we pull the two strands apart, and then that gives us two replication forks. So let's grab a pen to mark this. Right. Okay, so here is one replication fork in here. And here is another replication fork in here. So at these points, we're going to start getting DNA replicated. And one piece of DNA is going to get replicated in one direction, remember, and the other is going to get replicated in the other direction. So if you think about what is happening at this fork, one of these pieces of DNA is getting replicated in one direction, and the other piece of DNA is getting replicated in the opposite direction. So one piece of DNA is always going away from the fork, and one is always going towards the fork. And the same thing is true over here. The DNA here is getting replicated in this direction, towards the fork, but on the other strand, it's going away from the fork. So this, as you can imagine, creates some complications at the actual replication fork. So we'll revisit that. Let's take a look and see what's actually happening in each of these steps. So to unwind the DNA, you have to have this enzyme called DNA topoisomerase. Here it is, and it works along with this enzyme called DNA helicase. So the helicase enzyme actually does the breaking of the hydrogen bonds and the pulling apart of the two strands. Now, if you imagine that you've got a helix wound around, two strands of DNA forming a double helix, and you start pulling those strands apart, the helix very rapidly starts getting twisted and knotted up. And so this enzyme, DNA topoisomerase, basically works with helicase to relieve all the knotting that happens and the twisting and the torquing that you get when you try and pull two strands apart. And you can try this. Get a couple of pieces of string and wrap them around one another. Have a friend hold one end, and then you pull the two strands back apart again. And what you'll see is things start to loop around, they get twisted, they form knots, that kind of thing, they get tangled. And so topoisomerase basically fixes that problem. Once the two strands are pulled apart, all of these proteins here in this kind of orange color basically cover the strands of DNA and stop them coming back together again. Because DNA, once you pull it apart, it's just likely to snap back together again because the bases match up. So these orange proteins here basically keep the two strands apart. They're called single-stranded binding proteins. And as DNA gets replicated, they get displaced from the DNA. So that's what happens when we unwind the DNA. Let's look at the actual chemistry of replication now. This is a little bit complicated. We touched on it a little bit earlier. Remember the first really important rule about DNA replication. You must always add to the 3' end of a strand of DNA. You must always add nucleotide to the 3' end. And so that means we're always going to make the new DNA 5' to 3'. So here is a piece of DNA over here on the left-hand side of the slide. Here's the template strand. This contains the information which we are going to use to make the new strand. When I say information, I mean the sequence of nucleotides. So the sequence of nucleotides here is C, A, T, G, T. Okay, here is the 3' end of the piece of DNA that we're going to use as a template, and here is the 5' end down here at the bottom. Now remember, it doesn't matter whether you draw this upside down, left to right, diagonal, anything, as long as you've got the chemical polarity on here. So remember, the DNA molecules have to be anti-parallel. The two strands are the same distance apart, but they run in opposite directions. So if in our diagram on the screen, the parent or the template strand is running 5' to 3' bottom to top as we're looking at this, the other strand must run 5' to 3' in the other direction, so down the screen as we're looking at it. So here is the 5' end of the new strand, the daughter strand, and we've got to add a nucleotide to this end down here, to the 3' end. So the template strand has a G. That's the information. And so what do we put opposite a G? We put a C, everybody knows that. So here is our C nucleotide that's going to be coming in to base pair with the G, and you can see it's coming in with these 3 phosphates. And so the enzymes that actually do the business of linking all these nucleotides together using condensation chemistry is basically going to cut 2 of these phosphates off, and when that happens and we link this phosphate onto this hydroxyl here, that's the condensation chemistry. And when that new bond forms, the energy is released that is required to do the work of DNA replication. So what happens is these 2 phosphates get released. Those are the 2 phosphates over here. They get released. And as they get released and the new bond is formed between the phosphate that's left and the hydroxyl on the end nucleotide in the piece of DNA we're extending, work is done, and the new phosphodiester bond is formed. So here is the new phosphodiester bond that we just formed, and now we've got a C base paired with a G. So the next nucleotide would come in here, and its 3 phosphates would attack the 3' hydroxyl, and the same thing would happen. So that's a lot of complicated chemistry that we're not going to get into, but it is important you understand it to this kind of level. So let's now start talking about how we actually do the business of making the 2 strands in a way that one runs towards the replication fork and one runs away from the replication fork. So here's a piece of DNA, and we're not looking at it in the helix form just to make things simpler. Let's get a pen. Okay, so this end of the template must be the 3' end, and down here must be the 5' end. So let's label those. And I know that because this new piece of DNA is 5' at this end and 3' down here. So this is a new piece of nucleic acid being formed. And I was very careful there to say nucleic acid because in DNA replication, the first piece of nucleic acid that is actually made is not DNA. It's actually a short piece of RNA. And this is because the enzymes that do this, primarily DNA polymerase, it can't just start linking nucleotides together but there's an enzyme called DNA primase, sometimes called RNA primase, we're going to call it DNA primase, that can do that. So DNA polymerase, which actually does all the DNA replication, can't just start from scratch. It can't just start de novo, as we say. It has to have a free 3' end, a nucleotide already in place, so it can add to the end of that. And so the way that the cell overcomes this is it basically makes a short piece of RNA. And so DNA primase makes this RNA primer, as it's called, a short sequence of nucleotides, 10 to 20 nucleotides usually, and that lets DNA polymerase start making DNA. So if we take a look at what's going to happen, the next nucleotide would come in here on the 3' end, the next one would come in here, still on the 3' end. Direction of synthesis would be right to left as it's drawn on the screen. Now, we would be going in the other direction, away from the replication fork, when we work on the other strand. You must still make this RNA primer because DNA polymerase can't just start from scratch. So there'll be a short RNA primer on the other strand, and then you'd have DNA getting made. So let's put this into the context of a double helix and a replication fork. Let's just take a look at this replication fork on the left here. So let's take a look at this replication fork here and blow that up and take a look to see what's going on. And you've got to remember the same thing is going to be happening over here, and we'll come back to that in a moment. So what is going on in this replication fork here? So what we have is our short little RNA primer that's being made here, and the information in the template is being read from right to left. So here's an important point. You always make nucleic acids 5' to 3', but you read the information in the template 3' to 5'. So as this piece of DNA is being made right to left of it as it's being drawn, the information is being read chemically in the other direction, 3' to 5'. So that's an important distinction to make between the direction in which you read information and the direction in which you synthesize new DNA. So we've got this top strand here, this new daughter strand being made from right to left as we see, and that's 5' to 3', as it must always be. Now, because of the anti-parallel nature of DNA, we must be making the other strand moving away from the replication fork. So here's the problem. The enzymes that do all of the DNA synthesis sit right in here in the replication fork. And so that's easy because as long as we keep unwinding the double helix, going from right to left as it's drawn here, as long as we keep unwinding the DNA double helix in the direction right to left as we're looking at it, we can keep the enzymes in the middle of the replication fork, always adding new nucleotides onto the 3' end of this top strand, and that top strand is called the leading strand. But the problem is that this 3' end is going to be kind of running away from the double helix. And so that's a problem, running away from the replication fork. So that's a problem for synthesis that has to be overcome. So here's how that gets overcome. The top fragment of DNA that we're looking at it here, so remember this is called the leading strand, sometimes called the continuously synthesized strand. So the continuously synthesized strand just keeps getting made constantly because the replication machinery sits in here. And so the 3' end is always available. The way that the cell overcomes this other strand being synthesized away from the replication fork is it's made in lots of little short fragments, and those fragments are called Okazaki fragments after the famous Japanese scientist that discovered them. And so what happens is this first fragment gets made, and then the replication fork makes a second fragment here, and then those two fragments will ultimately get joined up by some enzymes that work to link them two together, and then a new fragment, a third fragment, will get made at the replication fork, and these two are being linked together, and then shortly these two will be linked together as well. And so then a fourth fragment, as the replication fork moves from right to left as we're looking at it, as the double helix gets unwound, a new fragment will get made in here, and eventually these two will get linked up. So the leading strand is made continuously, that's why it's sometimes called the continuous strand, and the lagging strand is sometimes called the discontinuous strand because it's made in these series of fragments. That is very difficult to visualize on a static image, so it's critically important that this is what you look at in some videos that are posted in Canvas. So check those out. So let's just review some of the language and the enzymes and things and look to see what's going on. Remember, the first thing that we always make when we do this DNA synthesis business is a short piece of RNA using this enzyme DNA primase, and that is true on both the leading strand and the lagging strand. Remember, the leading strand goes towards the fork and the lagging strand comes away from the fork, and that is because DNA is anti-parallel. So the next thing that happens is that we make the actual DNA, and so here the leading strand, the continuous strand, is made by this enzyme DNA polymerase 3, and that is continuously made. The helix keeps unwinding, and so it keeps moving in the direction right to left as we've shown it. But bear in mind, this is shown here as the fork moving right to left. We could twist it up, we could twist it down, we could rotate it around, and so we always refer to these in some kind of direction, just as a point of reference. So I'm just saying right to left so you can follow along with what I'm talking about. Excuse me, I rub my eye. Okay, so same thing happens on the other strand. Here is the RNA primer that's being made in the second Okazaki fragment. Here is our first Okazaki fragment, and here is our second Okazaki fragment that is now being synthesized. And then they will get joined together. So if we go on forward one step, if you like, here is our leading strand still being made continuously. Here is our leading strand constantly being made towards the replication fork. Here is our first and second Okazaki fragments being joined together now. And now we're making a third Okazaki fragment at the replication fork. And as that DNA polymerase moves away from the replication fork, another Okazaki fragment will be started at the fork. So there's this kind of like stepping backwards process. And this is pretty complicated, and there's a very unique, unusual structure which happens so that this can all work out in terms of holding the DNA in the right conformation for this to happen. So that's pretty complicated. That mechanism of DNA replication is said to be semi-conservative. Books often talk about some other models as well, conservative models and dispersive models. I don't tend to get into those because I want you to learn the correct one. It's called semi-conservative DNA replication because you start off with two original strands of DNA, the parental strands, the template strands. You separate them and you make some new strands. In each new daughter molecule that you get, you've got an old strand and a new strand, and so that's why it's called semi-conservative, because half of the original information is conserved, half semi, in each of the new daughter molecules you produce. So that's DNA replication very, very quickly. It's complicated. You must watch some videos, and I will see you again for our next section where we're going to take a look at chromosome structure.